So hello, everybody. My name is Karen, and I'm the manager for Plant-Ditech. It's a company from Israel that is a spin-off from the Hebrew University. And we developed a phenotyping system. I will share with you the slides. Okay. And the way we will build the session is that I will provide a certain short introduction about the system and its methodology. And then we have with us also Professor Menachem Moshelion, who will be talking about some examples, the technology and some examples. Menachem was the co-developer of the technology at the Hebrew University, and he will present later on some examples and use cases, different use cases of the system. Okay, so we shall start. So the principle of the system is very similar to a smartwatch. Similar principle of dynamic measurement of the physiological response in smartwatches from our body, but we do it for plants. So we measure the plant dynamic physiological response. We measure different traits that correlate highly to yield. So we can, for example, transpiration and stomata activity, stomatal conductance, biomass gain, and all of those, with the help of those, we can assess very quickly, within 1 month, more or less, instead of full seasons, we can assess the plant response to different stresses. We can then shorten the time dramatically for research in general, and also for seed development or any biology or biological material or chemical material. So with this platform, what we do, we measure and analyze all automatically the plant. So we don't touch the plant from the start of the experiment. The researchers is actually hands-free, and everything is being done automatically, handled by the system. So the control of each and every plant is controlled separately, and can get different cocktail of soil and water conditions. So each one can get its own soil and water conditions, and while we control the conditions with the system and the software, the software also take measurement with the help of sensors for the physiological status and the environmental status of the plant. And this happens dynamically about 500 times a day, so all day long. So we can capture very small differences in the plant response very quickly. With this, we analyze also in real time the results from the dynamic measurements of the plant in a comparative way. So we compare all the plants. So very quickly and also very precisely, we can get the different responses from the plant in very short time, within weeks. A little bit about the company. So we are from Israel. We are commercial since 2017. It was developed, as I said, at the Hebrew University, and we're a spin-off from that. We have distinguished customers in our install base. You can see some of the names here, like Wageningen, Donald Dadford in the US. The Wageningen is in Europe, the #1 university in agriculture. Mars Wrigley from the food sector, the cocoa plant breeder, but also the manufacturer of M&Ms and other chocolates. Weizmann Institute in Israel, Florida University in the States, the top 6. And then different also chemical and biochemical companies. So these are the type of customers we have. We have many more. This is just examples. We also have customers in China, And we will give some examples also of research done in China with this respect. This is the system, how it looks at Wageningen. You can see a large-scale system, and we will explain later on exactly how this works. This is in Mars Wrigley, just to give you a sense how the system looks with different plants and also different applications. The main applications for the system, so first is abiotic stress screening, because we can control the soil water conditions. We can also control flooding. So for example, for rice experiment and also for other purposes like flood stress, we also do that. We also provide physiological studies with the system, nutrients and bioag screening of different kinds, early detection of biotic stress, and Menachem will show examples for everything I described. Also seed treatments of how the whole plant is functioning after the application of seed treatments, and optimization of growth practices for plants. We work with many different plant types, from Arabidopsis to trees. You can see even trees on the system. So basically every type of plant is relevant, and also very easy to move from one plant to the other with no need to calibrate between different species. We have I will talk a little bit about also how we actually different from what is out there in terms of automation for phenotyping. So most of the automation you probably know is imaging. Imaging is very good when we talk about structure and color, and provides this kind of information. It's also slow in its response because the plant takes time until it develops a different structure. Also, you cannot measure many plants at the same time. You have to measure once a day, twice a day max. And it's also harder to analyze in terms of understanding what the image is actually, how the image is being analyzed, and how to translate the image analysis to data. And in our system, what we do, we again mimic the smartwatch in a way, because we are measuring the function of the plant, functionality, not exactly how it looks, but really about its function. We measure all the plants dynamically and simultaneously for immediate response, and very easy therefore to analyze because it's all quantified immediately. No image is taken, it's all based on sensors. And we have a lot of also control abilities, where in imaging it's more limited. This is just an example of such of a system where you can see the camera as well as the system below. So the lysimeter system below, this is our system. You can see all the green light taken. It's actually data taken. So this is the speed of data retrieval from the system. Very quickly, dynamic, all day long, where the camera moves very, very slowly and takes only a few plants at a time. We take all of this 200 in this example at a time. This is the system at Wageningen. Okay. Okay. I will now move to Menachem. Menachem will describe the technology and also use cases, how it can be used for different research questions. So I will just stop sharing.
So hello everyone, my name is Dr. Menachem Oshelion. I'm from the Faculty of Agriculture at the Hebrew University of Jerusalem. I'm going to explain you about the technology, how the system works. So first of all, the system could be arranged in any greenhouse, any growth room. It's very simple to install. And for each pot, you have the controller. This gives you a lot of benefits I will explain later. One of the best benefits, you can really build a randomized experiment. So each plant can get its own treatment. Doesn't matter where it sits in the greenhouse. So you can put as many solutions you want, and each plant going to get exactly what you want it to get. So this is the controller. The controller controls the irrigation system. So you can actually irrigate each plant in 2 valves, 1 and 2. So each plant can get 2 valves. The controller also connected to soil sensor, to atmospheric sensor, and of course to the lazy meter itself. I will soon explain all of this plastic, which helps us to get a lot of, or a very good signal to noise ratio. So as I told you, one of the benefits that we can irrigate with 2 valves, valve A and valve B, and this enables us to irrigate by time or by weight. So we can give a certain weight to each plant in grams, or by the plant own transpiration. So if the plant transpires 200 ml, we can bring back either 250 or 150, as much as we want per plant. So each plant in the array can get the irrigation in a feedback loop based on its own transpiration. And of course, we can also make the hydration rate. We can irrigate by the sensor, so we can choose the sensor and use the sensor to choose the irrigation pattern. So this is a very sophisticated system. This is the most sophisticated system today that enable you to irrigate as you wish, each plant separately. What more do we measure? So first we irrigate so we can give each plant its own treatment. It could be nutrient, it could be salinity, it could be drought, it could be different soils, different pHs, and many other aspects, including oxygen and EC and so on. So the system, the same controller you saw here, can be connected to many sensors, not just these 3, but many sensors. And the sensors could be pH, O2, water, EC, temperature in the soil, could be depth of the water, could measure root influx, could measure NDVI, infrared, PAR, humidity, temperature, and so on and so forth. So all of these, what I mentioned here, these are simple sensors. But the system can also measure stomatal conductance from the whole plant, like gas exchange do only for all the plant, I will explain later, the whole plant transpiration, the whole plant biomass gain, water use efficiency, and root influx. So everything you see here in green is physiological measurement that the system can measure simultaneously and continuously to all the plant in the greenhouse, few hundreds times per day, per plant per day. Why do we need this resolution? Because if you work in a non-controlled greenhouse, you have all the time changes of temperature, humidity, light, and so on, and you want to measure the plant respond to the real life condition. This is why we call it functional phenotyping. So the plant is standing in the middle of our of the experiment. The plant is exposed to all of this very variation, radiation, temperature, light. Everything is dynamic, right? Nothing is fixed in the greenhouse. From light to VPD to pH to EC, this is what the plant sends. Of course, these changes impact the plant's behavior, like stomatal conductance or transpiration, and this also make a lot of noise. Okay, if you just measure it, you will have, with a simple system like simple lazy meter, you're going to have temperature noises, you're going to have static noises, you're going to have vibration noises, you're going to have drainage from the pot, you're going to have evaporation. All of these are noises, but the Plant-Ditech technology know how to filter all these noises and give you a very clean signal. Okay, we call it high signal to noise ratio. Let me show you some bad example from the market. So other plant in the market, you see this drop here. So the plant just drop the water on the bottom. So you have a lot of water loss from the bottom or from the top. So you can see it in many other, these are not PlantArray system. You can see they lose water either from this plate, and they need to cover with all kind of plastic bags, they plants. This is another sample. So they put this bucket on the bottom, but you see a lot of water going to lose from this part. which you don't know if it's coming from the plant. You can also see this algae growing in the bottom. So this is a very bad systems. You can see these systems are not covered on the top or not covered on the bottom. This is another example. The plant just sitting on a table, a lot of vibration. This table is actually a vibration trap. So if somebody walk on the floor here, you will see vibration here. Not to mention that the lysimeter are exposed to direct sunlight. So the temperature of the lysimeter here could get to 60 degrees. And of course, it's fully exposed and drained. So these are very bad examples. PlantArray, from the other hand, has this very sophisticated, unique plastic that help you to prevent evaporation, to prevent excess water. You see, we have an O-ring for each plant. We have this green tab. These are patented that enable you, in the end, to protect all the noises I was talking about before and reduce all of these noises and enable you to see a very clean signal, including the irrigation, which you see it's irrigated all over the plant, not just in one spot like this system that irrigate only at one spot, so the root will grow only here. So the Plant-Ditech give you a fully surrounded irrigation for each plot. So the precision is very, very high with this system, which enable you even to measure Arabidopsis plant. It's in the resolution gram. All in all, when you grow your plants, let's look at this movie. This picture was taken in the greenhouse where each plant is getting different treatment, and we get the data. You can see the plant growing every day. Each picture is one day, and all the data coming clean directly to your software, via the software directly to your computer. So the nice thing is that you can get the data automatically coming to your system. So first, we control each pot separately, and second, we get all the data automatically. We also have a very good, sophisticated statistical tools. You can do online ANOVA t-tests and other tested in real time on your data. Because the system gives the treatment, so the system know which plant got which treatment, so you can get the grouping very fast and the statistical very fast. So you can get your data in real time, and this very much helpful because you can actually change treatments if you see they are not beneficial, or even stop the experiment and save time. Let's see some use cases. This study was published from Wageningen University. They have the system. In these systems, they measure our ability. As I show you here, we know to measure stomatal conductance. Stomatal conductance is a very tricky measurement, usually taken only by light course, gas exchange, porometers, and we do it automatically via the system. So Wageningen University decided to test our ability to measure stomatal conductance. So they measure stomatal conductance, and this is our system. They found this paper showed that our system give you 0.99 correlation to their manual measurement. So we got a very high ranking. This was published. Another interesting publish was just recently published in Cell paper, one of the best paper in the world. Here they show that plants can actually make sounds, and they monitor the plant's drought using our PlantArray system. So in black, you can see our data, and in red, their microphone data. These are two very different examples show you how unique our system, how accurate it can be, and how unique it could be integrated in a very different type of experiments. The thing in common that these experiments are always measuring physiological parameters. Another experiment coming from Professor Pei Xu in China. Here they measure cowpea, a legume, and they expose them different line to drought, and they could very nicely detect different type of plant response to drought. So the plant looked the same. There were, in the camera, they didn't see any changes, but when they were exposed to drought, they clearly found two different types. They actually find more than two, but these two were the most extreme of drought response. This was done, you can see, in less than a month. So in less than a month, you can screen your plant and select out the plants with the most desirable traits. And actually, there is almost no effort. You just put them on the system, and that's it. All the data coming to a computer. You don't need to send man and measure it on the system. Just get the data on the computer. Why this system is so important? Why is this regulation is so important? Because here the drought were feedback irrigated. If you do not control the drought, then you get a big variety. This experiment was done on tomato plants. Here the drought treatment was not controlled. And what you see here, that if you do not control drought, you get a big variation. So these are highly transpiring plant, and these are lower transpiring plant. This is a big problem. So in this paper, they could not separate between the tolerant to the non-tolerant drought plants because they could not control it properly as they should. In this experiment, they took the same tomato line. These are the IL line, introgression line developed by Professor Dani Zamir. But at this experiment, what we did, we exposed the plant to similar drought. So what you see here in this experiment, this is the daily transpiration. This experiment came to select different IL line, different tomato line, based on their physiological behavior. This line, 8.1, was found in the field to be a very poor drought-tolerant plant. And this plant, 5.2, was found in the field to be high tolerant plant. And what you see, that this plant actually developed a much faster transpiration. These are physiological traits, right? This is not coming from a camera. So you can see it day by day. These are day resolution. The green plant become much faster growing, but when you expose it to drought, you see the drought start here. Again, everything is automatically controlled by the system. So you see the soil water content is controlled, and the plant exposed to drought. So the drought is similar. It doesn't matter that this plant transpire more than this one. They are both exposed to the identical drought treatment and a recovery treatment. So you see the soil water content is identical to both of them, but the transpiration is very different. Look at the pattern of transpiration between these two lines. This line, for example, the green one, this is the recovery, much faster. What we found here, that the fast growing, fast closing of stomata, but fast recovery, actually is very important to high tolerant in the field, okay? This experiment was done in 1 month. In 1 month, we got a mechanistic explanation for why these plants are much better in the field. And this is intermediate plant. You can see it just in the middle of these two. And of course, you can screen literally tens of plants in this way, all of them simultaneously, and you find all of this statistic. All of this you see here was done automatically by the system. So you can really grow together, literally hundreds of plants together, and in real time select the ones that have much better trait for your desirable question. You can also look not on a day pattern, but on a momentarily pattern. This is a minute by minute. You can see that this plant has much better transpiration than the other at this time, while in the other times it's closed. And you can see this response that you see here directly to the light and to the VPD in red, light in black. So you can really see the plant different response minute by minute. So you can get a very, very high resolution, which is very important to get a mechanistical explanation. If you take this plant and you go to the field, you can find a very good correlation. This was published. This is together with Professor Dani Zamir. You can see a field experiment. This was published, so you can get the paper. A very good correlation between what we found on the system and the real yield. So the system prediction to yield is very, very high. And I want to remind you, we do this in less than a month or in a month's period. So in a month's period, you can get a very good prediction of the plant performance in the field. This is another paper showing this correlation, this time on wheat, also published. This is on pine trees, Pinus alpinensis. So here we grow the plants, big plants in big pots. And here we want to extract metabolite, hormones and sugars, but we want to make sure the plant suffered the exact same stress. So the point we extract the metabolite, you see it here, was based on the plant stomatal conductance. As I told you, our measurement is very accurate. So when you collect your RNA based on RNA and metabolites, based on the stomatal conductance, actually the result was amazing. You can see the paper, it was also published. This enables you to collect data precisely under the same stress. So if you want to compare different plants to the same stress and get RNA or DNA or metabolite analysis, and you don't do it with the system, you're going to get a lot of noise. But if you do it using the system and collect it based on the plant physiological response, actually the data looks very good and give you very nice result. You can see this in this paper. Another example is working with biostimulant company. It's called ICL. It's a very big chemical company. Here's a user, their materials, their biostimulant, and you can see that after 1 week, we get significant difference. So this is their product, and this is the control. Each place you see a red point, it means significantly different. So their product could be proven to be better after about 10 days. Okay? In the field, this experiment would take 3 months to prove, if not more. So this shows you how efficient the physiological or the functional phenotyping. You can get a very nice comparative analysis very quickly, and this data translate to the field very efficiently. This is another experiment show you this time 3 biostimulant and 3 treatments. So you had actually 6 independent treatment. You can see they use seaweed here, and they use polysugars, and they use normal irrigation and control and drought. All of them could be arranged in a truly block randomized, so it's the best structure for this kind of experiment. And the result, I mean, all the experiment was fully automated under normal farming conditions. We can skip the details. It was published, so you can find this paper if you want here. Let me take you to the conclusions. The conclusion was that the seaweed improved the plant growth under normal irrigation. This is the normal irrigation. The new material that ICL developed, the polysugars, improved the growth under normal, but not as much as the seaweed. But under drought, the new material actually prolonged the drought to protect the plant much, much better than the control and the seaweed. And the recovery, what we call resilient. Resilient is the recovery rate of the plant after stress. The resilient was much better using this material. Again, all of this data was established in about 35 days. So in 1.5 months, you can get a very complex system of 3 treatment, drought, recovery. In this experience, there were 72 plants simultaneously, and the data is very, very robust. This data was later proven also in the field. Another experiment, this shows you a combination of salinity and potassium. So here we mixed 6 different cell lines, 3 concentration of potassium, 3 concentration of salt, and we follow, the research question was, would potassium increase will help the plant protection to a higher NaCl? And so we used 3 different of potassium and 3 different of NaCl. And the answer was yes, higher potassium actually protect the plant from NaCl toxicity. Again, relative short experiment, and in few weeks we could prove this efficient potassium concentration that was later implemented for the farmer. Like in half a year, the result could be implemented in the farming. So it's a very, very quick decision system. Another novel aspect is disease finding. If you have fusarium or any other root disease, it takes a lot of time until you see something in your eyes. You can see this plant has fusarium in the roots, but this plant was taking picture here. You don't see anything, but when you take the root, you see a very black roots. This data come from the system, and you see that our system could distinguish this disease already at day 19, where you saw nothing in your eyes. So because these are the root disease, and the system measures transpiration, and we have a very good signal to noise ratio, the system detects these noises much, much before you see anything with your eyes. Actually, this paper is now in the bioarchive, so you can look for the paper and you will see we did it with many plants and many fusariums. it really works nice. If you work on disease, this can help you speed up your research much quicker than any phenotype you can see in your eyes. This is another fusarium. As I told you, we did it with several fusarium. This was done on pepper. This was done on tomato. We tried different kind of tomato. And you can see very quickly response of the plants to the fusarium, much before you see anything with your eyes. Similar result we got with nematode. Again, nematode is a big problem in agriculture, and if you want to develop some pesticide, again, nematode, you have to have a good testing, and you don't want to take out your roots. Here we show that using our system, we can detect nematodes in 2 weeks, very similar to the diseases. So biotic stress could be very, very easily detected by our system, as well as abiotic stress. Another thing, we can very easily integrate our system with many kind of cameras. So you can put your cameras above the canopy, take measurement of hyperspectral, RGB, infrared, and any other camera you want. And we have API in the system that enable you to integrate your data from your images into the physiological data that the system gives you. This paper also published, actually 2 papers like this already published. We will gladly send you these papers. Another approach of the system is to compare different soil types. So this is the plant transpiration based on the different soil types. You can easily see the differences in your eyes. Again, in like 3-4 weeks, you can get the optimum soil or nutrient that you need to your experiment. The system could be also integrated outdoors. So you see this system is actually in the open field, and these are wheat plants. You can see how nicely they grow. And in this experiment, they mimic drought experiment for this spot, and they grow next to these plants. The similar plant was growing in the field, and we can compare the plants to the real field condition. These plants are literally growing in the field, exposed to field condition, and the system is very robust and enable you to follow this experiment. These are another picture showing this field facility. In this system, you can really walk. These pots are 200 kilos. And you can see the plants are looking fantastic. So you can really grow trees, banana trees, wheat plants, whatever you want in the system, and get all the benefits I talked before of high sensor sensitivity, very good signal to noise, very sophisticated irrigation, drought response, nutrient, etc. So to conclude, the key benefits are fully automatic, that control and phenotype and analyze your experiment. You get many proven physiological traits. You get it in real time. You don't need to do another analysis. The analysis get to you automatically. It's not like image analysis that you have to analyze your data. You just get your data directly to the cloud. We have dynamic and simultaneous measurement. As I told you, hundreds of measurements per plant per day. And the environment. So we have the plant and environment, including soil, almost 500 times per day. You can shorten your test cycle. As I told you, months to 6 weeks is our average time to get a very significant result. So it's a very quick test cycle. We have a lot of papers, I think close to 50 papers in Plant Phys, Plant Cell, Plant Journal, Cell, PNAS, the best journals. So you can look it up. We can send you these papers. High correlation to field data. So we can really predict yield based on a real functional trait and not just algorithm that was done in model based. So it's a real simulation. We have multi-treatment that I explained. We have personalized. So each plant can get a different treatment automatically. You don't need to do anything. You just feed it to the system. We detect the immediate plant response, and I talk about minutes. So in 3 minutes, we see the plant respond. We see it much earlier than any other devices, and I talk about weeks earlier. And you get a real-time statistical analysis. So because we get the data phenotype in a real time, we can also get the statistic in a real time. And it's very modular and robust, so you can move the system, you can place them in different spaces, different tables, different growth chambers, in the field, wherever actually you want. Okay. I will be gladly now take your questions if you have any.